virus fit into that picture?
Well, it's a little bit challenging to talk about virus because you want to be careful about exactly what it is that you're talking about it. So there-
No
there's like,
kidding.
there's like, the virus, which means poison, but even before there was ever an electron microscope where they could possibly see what they're calling a virus these days, they already blamed a virus on several illnesses,
Right.
specifically ones that they couldn't find bacteria
Yeah.
associated with, like polio.
Oh, okay.
Right. So there's one thing like that where there's like this kind of virus hunt by medical researchers trying to find something that causes diseases that they don't have another explanation that they're looking at. But then there's also what has come out of the bacteriology research with bacteriophages. And bacteriophages, or bacteria eaters, they're called, and I think that term is actually a misnomer, is considered a model for viruses. And in fact, the original genetic sequences of viral genes were garnered from bacteriophages, because bacteriophages, I think most objectively, exist when you have artificial conditions on bacteria mostly. So you were talking about how, bacteria exist in nature. And one of the things is you never see an isolated variety of bacteria, species of bacteria in nature. There are always dozens or even hundreds of different types of bacteria mixed together, growing right in the same community, and they all exchange genetic information with each other. It's really only in the laboratory that scientists separate out the species and try to grow a pure culture of bacteria. And they're capable of doing this, but the bacteria are not healthy. They're under stress in this situation. But if you add more stress to the situation, what you'll see is that you'll see the presence of these phages in the bacterial culture, and you'll see the cells disappearing. But the scientists that originally discovered this and called them
When you say the cells disappear, you mean the bacterial cells disappear?
bacterial cells disappear and phages appear to be eating them.
Yeah.
But I think that this was a bit misinterpreted, because one of the main aspects is that in the modern mainstream medical science, they only look at dead things under a microscope. They don't look at living things. And when you observe dead things, it's very hard to take those observations and be sure that you can describe behavior that resulted in that. I mean, imagine trying to, learn about human ritual behavior from examining cadavers of humans.
Right. All right.
It'd be impossible.
Right.
So
I often say that the fundamental problem with Western science was we wanted to learn how to help frogs. So the first thing we did was kill the frog.
Yes, exactly.
Which I can guarantee did not help that frog.
It's funny, these lapses in logic and common sense sometimes.
Yeah, right.
But so if you look at the dead bacteria under the microscope, you can see, there's less bacterial cells over time and more phages. They must be consuming the bacteria and reproducing. But another explanation is that they actually turn into the phages.
Yeah,
now,
disintegrating.
Right. Well, when bacteria are under environmental stress in nature, when they're in a mixed culture, they turn into spores, generally speaking. And the spores can withstand the harsh environmental conditions. And then when they're right, the conditions return to normal, then they revert back into a living, growing state. And when they're in the laboratory under artificial conditions, when the bacteria are stressed even more, they can be induced into phages. But the bacteriophages are interesting scientifically because one is they've actually been shown clearly to exist. So they take a bacterial culture, do certain things to induce phages, and then they can put it in a centrifuge and separate the bacterial cells from the phages, and then look under a microscope, an electron microscope, because you need that level of magnification, and see just wall-to-wall phages in your sample, so it's pure. And another interesting thing is they have a very characteristic geometrical shape. So they're not spherical particles. they are actually structured particles, and they look a little bit like the Space Needle in Seattle. But on Stanford website, they have great pictures if anyone wants to look for themselves. But since these are pure particles, you can then basically characterize what chemicals are in the particles, because you know it's only coming from that since it's pure. So you can like, lyse the membrane and take the genetic material out, and then it'll be in one intact piece. And you sequence it end to end, and then say this is the genome of this, if it has a genome, and then you can look at the proteins in the membrane, or how the membrane, you can do further, chemical tests on it, so you can know exactly how that thing is structured and what it is. And so they've been able to do this kind of scientific methodology for other, what they call viruses that are in single-celled organisms like amoeba and sea algae. Are some other examples, and many different species of bacteria, not just one. Like, there are many different types of bacteriophages. I've even worked with them in the laboratory when I was in college.
Yeah.
So it's a pretty common thing. So,
and I think your point there is this idea that you sometimes hear, well, it's so complicated to isolate, purify, and characterize, is actually not true.
No, not at all.
It's just part of normal bacterial phage research. That's what you do.
Even before they could look under the microscope, they already were able to separate out what they thought were viruses from bacteria and other organisms by filtering.
Yeah.
And, you can go back to turn of the century papers on viruses, and they basically, took some diseased tissue from a person or an animal and filtered all the cells out, and then then it was basically toxic, and it could, if they gave it to some other animal the right way, it could sometimes cause illness. And so they said, well, there must be a virus in there, right? But they didn't know what it was, but they knew they would separate it out. So the first thing you could do is just put a sample through a filter, because virus particles, or what they say they are, is so much smaller than any other type of cell, right? You could separate it from any other type of cell, right? Bacteria, fungi, mammal cells, human cells, just by filtering it. So that would be an obvious thing that you could do. And that's not full purification, but this is something that you could do in an hour or two in the laboratory. It's not complicated, right? We've filtered things in our kitchen sometimes. But then the next step is, after you filter it, is that you do what they call density gradient centrifugation. And, centrifuge is just a fast spinning thing. You put it in a test tube in a medium that has a density gradient, because the spinning separates things by density. And all things of the same density will basically be in a little tight band on the test tube. And then you can take a syringe or a pipette and take that and then do experiments on it. And it may take a little practice, like how long do you run the centrifuge, which filter do you use? Do you filter more than once? Like, of course, all those things, it's harder when you're in the lab to do things than theoretically, but we're talking about technology that's been around for a long time. Yeah. Like, a centrifuge is not a new high-tech device, and neither is a filter, right? They were able to filter things over 100 years ago. So why aren't they doing these basic steps when they do science related to viruses that they say cause disease? And they did try to do that when they started off, but what happened is that what they observed was there was no predominant particle that was there that was identical to itself. There were mixtures of all kinds of debris and particles. And they basically were about to give up before they got to these tissue culture experiments. And I think you wanted to ask me about that, so I'll stop there.
Yeah, but I think again, correct me if I'm wrong, but my understanding is one of the other things that I know Stefan Lenka always talks about that virology disproved itself is they did learn to filter and isolate and purify. And they had cultures, maybe not of a pure single virus, but at least pure, quote, viruses or these bacteriophages. And then they would introduce those to normal animals, who weren't sick, and they couldn't make any of them sick. I mean, it was like, all right, we did it. We did it the way we're supposed to. We have the thing we're looking for. We squirted it down a hamster and monkeys and breathed into people's mouth. Nothing happened. Let's go home and be farmers.
Listen, with polio, it's unbelievable how
Yeah,
they
right.
tried, right, to make an animal sick. They took, basically dead babies who died of polio and ground up their brain and spinal cord in a blender and, gave it to all these monkeys. And eventually they had to inject it directly into the monkey's brain to
Brain.
make
Yeah.
them sick. But if you inject anything into a monkey's brain, it'll make
Right.
it sick.
Right. They might herniate their brain stem. Yeah.
But like we didn't mention the word exosome today, but right, exosomes are also small particles that are about the same size as they say viruses are, and they're made by our own cells. But when the same researchers that are unable to purify virus particles can purify exosome particles, no problem.
Yeah. Right.
Like you look at those papers, they do the same procedure I described with filtering and centrifuge that they did for bacteriophages, and they're able to characterize the exosomes very well because they have a pure sample of it.
And I happen to read the minutes of the 1973 Pasteur Institute conference, and they actually outlined the steps for how you would know that there's a new virus. And it's basically exactly what you said: filter and centrifuge and purify and characterize. And it was very clear and doable, and they just don't do it. So with that, how do they do a viral culture?
Well, it's very interesting and quite astonishing. But what they do is they take basically the raw sample from the sick person. So like, they want to look at a virus that supposedly makes someone sick, right? So they have a sick person, and depending on what part of their body is sick, they take a tissue or fluid from that part of the body. So like, let's say it causes a lung infection, they would take lung fluid, okay? And that is a very dirty sample because it has many, many things in it, many different kinds of cells, many different chemicals floating around in it, genetic material, etc. They don't filter that. What they do is they add that to a cell culture, and the cell culture cells are generally not human cells. They are sometimes. There was one paper I saw where they used lung cancer cells from a human. Right. But they're usually monkey kidney cells. That's the most common one. But these are like commercially available cell cultures, right, that you could just buy.
Egg yolk stuff. Yeah.
Yeah, you could culture these things on egg yolk probably. But what they do in these virus experiments is that they use minimal growth media. So in other words, just the basic amount of nutrition that the cells need to survive. Right. Not thriving type level of nutrition. Right. Then they add usually bovine calf serum. So like the blood product, the non-cellular blood product from a baby cow, which could have lots of things, foreign proteins in it that could cause damage to a cell. Then they add antibiotics. And they don't always specify which ones they add, but the reason is they say, well, they don't want any, bacteria to contaminate the sample. Right. But I thought this was really interesting, Tom, because you're familiar with amphotericin B, right? Yes. Fungal. So, amphoterrible, exactly, right? And we know it's very toxic, and it's like if someone's about to die is when they get amphotericin B. And it's an antifungal antibiotic. And one of the main toxicities it has is to the kidney. Well, they mixed it with kidney cells. So what do you think might happen there, right? So all the antibiotics that they mix, they're all toxic to cells. Like antibiotic means against life. Yeah. And they're also, antibiotics are known to induce cells to make exosomes. Right. So there's the calf serum, there's the minimal nutrition, and then there's the toxic antibiotics, and there may be some other things like some buffers and things like that. But that's the main recipe. And then they add in this contaminated lung fluid from someone who's got pneumonia, in with that cell culture. Now, then they observe the cell culture for several days, and they're looking for what they call cytopathic effects. So in other words, non-specific damage to the cells. Now, of course, if you mix a cell culture with a bunch of toxic things, and by the way, there are many things that could be toxic in that lung fluid as well,
Yes.
from a sick individual. It's not that surprising that you'll see damage to the cells. And when cells undergo damage, they behave in predictable ways. So early on, they would basically send out alarm signals. And one of those would be by putting out exosomes. And we know in these cell cultures, they're always going to do that because we gave them antibiotics. And we know from other research that antibiotics plus cells gives you exosomes.
Right.
And then when the cells start degrading, they basically put off all these kinds of vesicles, like they fragment. And this process can go, all the way to complete destruction of the cell. But the various different constituents get packaged up in the membrane as it pinches off and forms these little particles or vesicles. And this is a protective mechanism to the body, because if the cells released all the contents into your circulation, they would damage other parts of your body. Right. So containing them in these little vesicles as part of the dying process is a helpful thing. And basically what they do is that they look under the microscope at all of these particles in the damaged tissue, and they find one that they say is the virus. And then they show a picture of that. But what they don't do is they don't show pictures of the other particles or the exosomes and say, oh, here's where we found an exosome, and here's where we found the virus particle, and here's how we can tell one is one and one is the other. They also don't then take that cell culture mishmash and do the filtering and centrifuge and purify it out of that. So you never see like a picture from the microscope where the entire visual field of the slide is virus particles and they all look identical, right? In fact, even when they write about the sizes, right, sometimes they write that the particles range in size more than double, right? Imagine if, you say, well, okay, some humans are as short as 4 feet. Some humans are 6 feet and some humans are 12 feet, right? Range from 6 to 12 feet. Most animals don't have that much of a range in their, size. But all these things, it's like you'd expect to find something that is homogeneous,
right? Uniform,
right? And uniform, right? And that's not what they're seeing.
And to me, because as you know, I always think in analogies and metaphors, that the thing that's so interesting about the process of viral culture is the way that a viral culture is done to produce this cytopathic effect is basically starving and poisoning the tissue.
Absolutely.
Which is not unlike how people get sick. You starve, and in fact, it's like we learned, to me, what that proves is if you take a tissue and you want to damage it or kill it, or an organism, basically starve and poison it, and you got a really good shot at hurting it.
Exactly right. And I can just hear an indoctrinated virologist give you a counter argument. Well, we're just making the cell more vulnerable to the virus so that we can highlight the virus effects.
Yes, right. Except then why don't you do a control and just put saline and purified virus with a healthy, nourished, unpoisoned tissue and see what happens.
Yeah, well, that's an excellent point, and that is like Stefan Lanka right now is trying to raise funds to actually do a negative control experiment, and it's not trivial because you'd have to do really 2 negative controls. One where you add saline instead of the lung fluid, but there should be another one where you have the lung fluid of a healthy individual. That you add to the culture.
Yeah. All right, let's finish with one more thing, which is you hear a lot about, yes, but they have the entire genome of this virus. And first of all, I mean, if you don't again, I have an analogy. It's like if I said, Andy, there's a flying saucer over your head right now. And you might say, Tom, how do you know that? And I would say, well, I hear a whooshing noise through my microphone here. And you would say, well, that doesn't actually prove it. I was thinking you might say, I see it over your head. Well,
Right.
no, I don't see it. But I do see these little pieces of iron filing under your chair. And even though I've never actually seen or analyzed a flying saucer, I know that those iron filings could have only come from a flying saucer. And you might think I'm a lunatic because how do you know they didn't come from the chair or an airplane or something like that?
So, if to take that to the next logical step, right? So if you already characterized flying saucers and knew that they existed and were able to study their structure, right? And said that there's a specific iron alloy that they use, right? Then you can look at these iron filings and say, this is the specific flying saucer iron alloy, right?
Right.
But absolutely correct what you said. You can't take the iron filings and say, from this, I prove that flying saucers exist. And by trying to say that the genome proves the virus exists, that's exactly what you're doing. Because, well, where did this genome come from? If you didn't prove that there's a virus first, where did you get the genome from? So, it has to go in that logical progression. But they bypass that completely. And they do this in such a way. So like they might say that the genome sequence is discovered or that there is a genome for a virus. But what would be a much more accurate description would be that they discovered a theoretical computer model of a genome of a theoretical alleged virus.
Yes.
Right? That would be accurate to describe it, because what they do is, like you'd say, like, let's think, how did they do the Human Genome Project? Right? Now, they didn't mix humans with hundreds of other species of animals and put them in a blender and take out little fragments of DNA and put it together and say, these are human, right? What they did is we knew what a human was, and then we had a pure human and we took out some material from that pure human, and it was still contaminated, right? But we did some steps to make sure, like, because we tested from 100 other humans and we found the same thing in each one, for example, right? Or we validated it in some way. But what they're doing with with viral genomes is they're taking that sample, like I discussed with the tissue culture, like a lung fluid sample, and they're pre-identifying viral sequences. So they say, okay, this is a coronavirus, or this is a rhinovirus, or this is, a lentivirus, whatever they think they're looking for. They go to a computer database, which contains other sequences that they've determined in the same manner, by the way.
Yeah.
And they develop small PCR probes from those sequences, so that from a library. Basically, they say, okay, we know this is a sequence from another similar virus. So let's find if this sequence is present in this lung fluid sample. And what they do is they take dozens or maybe hundreds of these primers to start with. And then whatever they identify, they pull out little fragments that have that part of the sequence and then have another sequence after it, right? And these fragments might be 200 to 300 bases long. And then they sequence those. And then they make primers based on those sequences to find, like, well, a piece that would attach to it, right? And they make a whole, what they call a cDNA library of fragments like this. Okay, now in one paper I looked at, they had over 20,000 fragments of genetic material. So very different from starting with an intact organism, pulling out the intact genome in one piece, or, in one chromosome pieces, depending on the organism, and then being able to know exactly how, you maybe can break it into smaller pieces for sequencing, but you know exactly what goes where because you started with the whole. Right. In this case, you're starting with pieces. So then so you have like 20,000 fragments that are 200 to 300 bases long, and the genome is allegedly 30,000 bases long. So a lot of these fragments, and there's overlap and there's gaps and such. And when they sequence all these fragments, then they put them into a computer program, and the computer program basically solves the puzzle and puts them into one piece.
Yeah.
But there's of course errors because there are gaps, right, where there's not a continuous sequence. Then there's also like, how do you put it together? How do you know which ends match up with which? You know, like even if you do a jigsaw puzzle, right, you know, there are some of those pieces that they are almost a perfect match, but when you put it in there, you got to use just a little bit of force, so you know it's not right, right? So like, how do you know that the computer's not doing that? How do they fill in the gaps? Well, they fill in the gaps, they go back to the library of sequence, try to find a sequence that's close, and then just extrapolate it in there, you know, just fill it in from basically their imagination. So this, then they have, you know, after all this number crunching, and I mean, they need a computer to do this, because if they had people trying to do this by hand, they'd probably be working for, you know, 10 years before they could put it together. But then they get something and they publish it and they say, this is the genome.
Yeah, got it. All right. I'm going to let you go. Any final words you have for our audience? Just a word of advice, encouragement, anything.
Well,
maybe
you know,
in the justice realm.
Well, you know, I always say, don't take my word for it, right? Because I may be a meticulous researcher, but I'm still prone to making mistakes. And so, you know, look at other sources, try to make your own decisions about things. I definitely think that's really important. And, you know, just want to emphasize that learning about this stuff and being open to new ideas is, I feel, is very critical at this point in time, because we're in the midst of major societal change at every level, right? And a lot of it is, I mean, almost all of it is very oppressive. And it's all rooted in this kind of medical manipulation. And so if we are stuck in a dogmatic view of our health and the medical system, we're going to continue to be vulnerable to this kind of, you know, medical tyranny, medical martial law, health technocracy, whatever word you want to use to describe it. And my goal is to really, you know, try to educate people so that they're not vulnerable to that way of being manipulated any longer, and, you know, allow them to basically, you know, stay as free people as much as possible.
Great. All right, Andy. I so appreciate you taking the time to talk with me today.
Well, it's definitely my pleasure, Tom, and congratulations on your new venture here, speaking to the people on your platform.
Yeah. Okay. And we will send you a copy of the new book, The Contagion Myth, as soon as it comes out.
Awesome.
All right, Andy. Take care.
All right. You too, Tom. Thanks.
Okay. Thank you.